Good afternoon to one and all present here. On behalf of the organizing committee and all attendees of the conference entitled Neoperspective in Biochemical, Chemical and Microbial Techniques, I extend a warm and heartfelt welcome to our esteemed speaker, Dr. Ramachandran Samivel, currently working as Assistant Professor in Research, Cornea Researcher, College of Applied Medical Sciences, King South University. He completed three postdoctoral researches, one from Dan Cook University, College of Medicine, South Korea, the second one from Tamil Nadu Veterinary and Animal Sciences University, India, the third one from Department of Physiology, Tulane University, USA. He is having totally 18 years of teaching experience. To his credit, he is having 25 research publications in various international journals with high impact factor. Sir, your expertise spans biology, microbiology, biology, and nanotechnology, and we are honored to have you to share your knowledge with us at this prestigious international conference. Your presence adds tremendous value to our event, and we are eagerly anticipating your address. As a distinguished expert in your field, your contribution will undoubtedly enrich our discussion and inspire all those in attendance. Once again, thank you for accepting our invitation and joining with us today. We are truly privileged to have you as a part of our event, and we look forward to your insightful address. The stage is yours, sir.
Thank you, Doctor.
ஏய் அன்மியூட் பண்ணாம பேசியிருக்கனா
Uh,
சார் நீங்க ஸ்டார்ட் பண்றீங்களா
yes, thank you, Doctor, for your warm welcome.
சார்
Uh,
are you there?
I'm yes, I am here. Are you hear my voice?
Yes sir,
Hello.
you're audible.
Yeah, yeah.
Hello sir, yes sir, you're audible sir, thank you.
Okay, okay. Thank you so much for your kind introduction. Today, I'm going to talk about antibiotic resistant and bacterial susceptibility and biofilm construction. Uh, this slides this is the outlines of my presentation. Especially, I'm going to discuss about structure and role of bacteria and its subcellular organelles, especially external and cell envelope, such as intermediate and internal organelles. Then I'm going to discuss about antibiotic resistant bacterial susceptibility and biofilm constructions. Third, I'm going to elaborately discuss about synthesis and characterizations of Nilavembu Choornam-gold nanoparticles and its synergistic effect of antibiotic resistance susceptibility. Yeah, this is the basic introductions of mm, s- um, eukaryotic and prokaryotic cells. First, I'm going to characterize characteristics of cell natures. All living things, especially single and multicellular organism, are made up of cells that share some common characters, especially basic shape, internal content, DNA chromosome. It is have two basic cell types, especially eukaryotic and prokaryotic. The eukaryotic cells, such as animals, plant, fungus, and protist, it contains membrane-bound organelles that compartmentalize the cytoplasm and perform specific functions. It also contains double membrane-bound nucleus with DNA chromosomes. Next, the prokaryotic cells, such as bacteria and organs, there is no nucleus or other membrane bound organelles. These are the prokaryotic and eukaryotic cells. Among these two cell types, They have some common subcellular organs, especially cell membrane or plasma membrane, cytoplasm, ribosome, and nucleoid that DNA coils. Yeah, my study especially going to elaborately discuss about prokaryotic cell profiles. That prokaryotic cells have three segments depending upon that locations. It's divided to external cell and envelopment. It means intermediate and internals. That external have uh, appendages is located flagella, filii, and fimbriae. Then it have glycocalyx. The glycocalyx have capsules and slim layers that in cell envelopes intermediate composed cell walls, cell membranes. That inner segment compose cytoplasmic matrix, ribosome, nucleations, and nucleotide chromosomes, actin cytoskeletons, and endospores. Yeah, this slide shows structure of bacterial cell walls. This is the horizontal cross section of prokaryotic cells. It have external, intermediate, and internal subcellular cellular organs. Uh, yeah, next slide. I'm going to elaborately discuss about external, intermediate, and internal subcellular organs. First, this is the external structures. As external structures have appendages. There are two major groups of appendages depending upon motility and attachments. The motility have flagella and axial filament. It is called periplasmic flagella. The next attachment or channel ha- uh, have divided into fibrae and fleas. Then third one, glycocalyx. This glycocalyx have surface coating, especially formations of capsule and slim layers. Yeah. Uh, this slide shows structure of flagella and its functions. Flagella have three parts: filament, hook, and basal bodies. Filaments have long, thin helical structure composed of proteins such called flagellins. That intermediate hook have curved sheath and basal body have stalk of ring firmly anchored in cell walls. It rotates a 360-degree. It have a number and arrangement of flagella varies from organism to organism. It's divided in four categories depending upon the flagella natures, especially monotrichous, lopotrichous, ambotrichous, peritrichous. Its main function is motility of cell through the environments. Yeah, this is the structure, 3D structure of flagella membrane channels. I already told you they have three segment: first filament, hook, and basal body. It anchor with cell membrane and cell walls. Yeah. Uh, this slide shows flagella arrangement and responses. Also, already mentioned about they have four categories. One is monotrichous. It have single filament at one end. This is the single end monotrichous. The lopotrichous have small bunch emerging from the same site. Yeah, this is the lopotrichous. The third one, amphotrichous, have flagella at both end of the cells. Yeah, this is the amphitrichous. Fourth one, peritrichus, it have flagella dispersed over surface of the cells. It move slowest and deeper. Yeah, it occur around the cell bodies. Okay, it guide bacteria in the direction in response to external stimuli such as chemical stimuli and light stimuli. Chemical stimuli mainly involved hemotoxis. It regulate positive and negative barriers, and light stimuli involve phototaxis. Yeah, yeah, that of second one, periplasmic flagella. It is internal flagella. It enclosed in the phase between the outer sheath and the cell wall peptidoglycans. It produces cell motility by contracting and importing, twisting, or flexing the motions. Yeah, this is the periplasmic flagella ultrastructure and cross sections. Yeah, this is the that this slide shows fimbriae. It is fine and proteases hair-like crystal emerging from the cell surface. Their main function in adherent to other cells and surface. The fourth one, villi. It is a rigid tubular structure made up of lean proteins. It found only in gram-negative bacteria. It function to join bacterial cells to partial DNA transfer called conjugations. Yeah, these are the please transmit DNA content and genetic substance. Yeah, this outer is called fimbriae. Yeah, that fourth one, glycocalyx, it mainly involved coating of molecules external to the cell walls. It also made up of sugars and proteins and other moieties. They have two types of galaxies. It's depending upon the coating. Uh, One is a slim layer and second one is capsules. That slim layer have loosely organized and attached. That capsule have highly organized and tightly attached. Yeah, this is the slim layer. This is the capsule coat. Okay, it main function is protect cells from the dehydration and nutrient loss, and also inhibit killing by white blood cells by phagocytosis, and also contribute to pathogenicity. It mainly involve attachment and formations of biofilms. Yeah, this is the flow graphical flow rate of that single bacteria colonies attached to biomaterials. Then it make glycocalyx coat formation. It transmit to biofilm formation and maturation. Then it make complex matrix formations with DNA, single stranded DNA, proteins, and lipopolysaccharides. Yeah, for these steps, let's indicate that for glycocalyx formation, it took 3 hours. After that, at 8th hour, it make complete glycocalyx with biofilm formation. Then uh, at 11th hour, it completely matured biofilm with that slim coating matrix. Yeah, yeah, this slide shows cell envelope such as intermediate layers. That intermediate layers have external covering outside the cytoplasm. It composed of two basic layers, cell walls and cell membranes. It mainly involve maintenance cell integrity. There are two different groups of bacteria demonstrated by Gram stains, such as gram positive bacteria and gram negative bacteria. Gram positive bacteria have thick cell wall composed primarily of peptidoglycan and cell membrane. Gram-negative bacteria have outer cell membrane, thin peptido layers, and cell membranes. Yeah, this is the Alstar structure of gram-positive and gram-negative bacterial cell walls. Yeah, here clearly indicate that gram-positive bacteria have thicker bacterial cell wall and cell membrane. Yeah, here that gram negative bacteria, it's clearly indicate that they have three layers: outer cell membrane and inner cell membrane, and like peptidoglycans layers. Yeah, this slide shows cell walls of gram-positive cell bacteria. It have very thick, approximately 20 to 80 nanometer thickness. It is homogeneous sheath of peptidoglycans. It include tecnoic acid and lipotecnoic acids. It function in cell wall maintenance and enlargement during cell divisions. It also move cation across the cell envelopes. It stimulate specific immune responses. Some cells have periplasmic phases between the cell membrane and the cell walls. Yeah. Next slide. This slide shows gram-negative bacterial cell walls. That gram-negative bacterial cell walls have thinner, approximately 8 to 11 nanometer thickness. It composed of an outer membrane and a thin peptidoglycan layers. Uh, the outer membrane is similar to cell membrane bilayer structure. Outermost layer contains lipopolysaccharides and lipoproteins. Mm, Further, that lipoportions, it's called endotoxin, may become toxic when released during the infections in the um, uh, foreign bodies. May function as act as receptors and blocking immune responses. It also contain porine proteins in upper layers. It regulates molecule entering and leaving the cells. Also, the bottom layer is a thin sheet of peptidoglycan. Uh, similarly, periplasmic phases between above and below peptidoglycan layers present. This is the sandwich alter structure of gram-positive and gram-negative bacteria. This gram-positive bacteria have thicker cell walls and cell membranes. And this gram-negative bacteria have three different layer of outer and inner layer and peptidoglycan layers. It composed with phosphomydes. Yeah, this slide shows composition comparisons of gram positive and gram negative bacterial cell walls. As I already have told you, these gram positive and gram negative bacteria varies the uh, inner subcellular moieties. Yeah, this slide shows cell membrane structures. Cell membrane structures have phospholipid bilayer with embedded proteins such as fluid mosaic models. Its functions are providing site for energy reactions, nutrient processing, and biosynthesis, and also passage of nutrient into the cells and the discharge of wastes. Also, cell membrane is selectively permeable to allow the substance. Yeah, this is the sandwich structure of cell membrane. It composed with lipid bilayer with integral protein, glycoproteins, and carbohydrate receptor and peripheral proteins complexes. Yeah, this slides showing internal structure of prokaryotic cells. That internal structure especially have cell cytoplasm. It is dense gelatinous solutions of sugars, amino acid, and salts. It have 70 to 80% water. It serves as a solvent for material used in all cell functions. Next one, the chromosome. Chromosome have single circular double stranded DNA molecules that contains all the genetic information recorded by a cells. It aggregated in a dense area called the nucleoid. Nucleoid DNA is tightly coiled. Mm, Third one, ribosomes. Ribosome made up of 60% muscle RNA and 40% protein moieties. It consists of two subunits, large and small subunits. That fourth one, endospores. Endospores have two types. One is two functional characterization. One is sporulation, and second one is germination. Sporulations, it's called formations of endospores, which stand extreme in heat, drying, freezing, radiation, and chemical substance. That germination, it turn to vegetative growth, and it multiplications of subcells. It mainly involve resistance linked to high levels of calcium and dipicolinic acids. It have dehydrated and metabolically inactive and thick coat. It is a longevity verges on immortality. It is stable to 50 million years. It also resistant to ordinary cleaning method and boiling temperature. It need functionalized pressurized steam at 120 degrees Celsius for 20 to 30 minutes. 30 minutes will destroy it. So according this introduction, uh, I am going to discuss about pathophysiological role of antibiotic resistant and biofilm constructions. Yeah, that antibiotic resistant is a serious public health problem in some bacteria that are capable to causing serious disease becoming resistant from the most commonly available antibiotics. That antibiotic resistant bacteria can spread from person to person in the community or from patient to patient in hospitalized peoples. Mm, it also antibiotics prescribed by health professional will failure during the emergence of multi-drug resistant bacteria strains. So this slide shows most antibiotic resistant bacteria strain. These given below organism were primarily isolated from bloodstream of post COVID-19 patients from China in 2009, end of 2019. Mm, Especially Klebsiella pneumonia, present in 8%, Staphylococcus aureus 10%, Pseudomonas aeruginosa 21%, uh, Stenotropomonas maltophilia 13%, Acinetobacter 22%. Most of these bacterial organisms proliferate during mono antibiotic resistant and multi antibiotic resistant, such as ceftriaxone, cells. These bacterial cells initiate attachment of biomaterial surface, it convert to biofilm secretions. It migrate to biofilm maturation. That biofilm maturation is make complex formations of bacterial cells with extracellular DNA, exopolysaccharides, proteins, and slim and glycocalyx matrix. This biofilm formation highly antibiotic resistance is may increase expressions of efflux pump. It means is resistance to antibiotics. So this complex again entered biofilm dispersion. Then it release plankotic cells. These plankotic cells again enter to another cycles. Yeah, this slide shows that biofilm mediated infection linked surface and tissue located biofilm pathogenicity. Okay, yeah, this is the nose. It's cause rhinosinusitis. And mouth it's cause chronic tonsils. In lung cause fibrosis, and liver it's cause biliary tract infections, and then urinary tract infections, kidney stone, and osteomyelitis in bone, and joint pains and inflammations, chronic wounds, and urinary catheter contaminations, endotracheal tube contamination, contact lens, its contamination, as well as cornea keratitis and dental plaque and endocarditis. Carditis, especially nowadays after post COVID pandemic, most of people suffered in cardiac injuries, as well as cystic fibrosis and kidney stones, because of that antibiotic resistant bacterial biofilms is deposited most of organs. This slide shows that biofilm mediated infections divided in two categories. One is device related biofilm infections and tissue related biofilm infections. Device related biofilm infections have uh, ventricular derivations and contact lenses, endotracheal tube, central vascular catheter, um, pacemaker catheter, cerebral vascular catheter, urinary catheter, hip prosthetic joints, orthopedic implants. That tissue related biofilm infections caused chronic rhinosinusitis, ear infection, dental plaque, endocarditis, cystic fibrosis infections, bloodstream infections, urinary tract infections, biliary tract infections, osteomyelitis in bone, chronic wounds. Yeah, according this biofilm, antibiotic resistant biofilm, here we are chose to four different categories of antibiotic resistant or non-resistant, gram positive and gram negative strains. Mm, Yeah, these tools are negative and gram positive antibiotic resistant strains, such as Klebsiella pneumonia and Staphylococcus aureus, and antibiotic non-resistant bacteria, gram positive and gram negative bacteria, such as Pseudomonas aeruginosa and Enterococcus faecalis. Yeah, this is the that device catheter surface have biofilm and glycocalyx slim bacterial contaminations. Yeah. Yeah, this is the also structure of antibiotic resistant gram positive and gram negative bacteria. Here is clearly shows between the bacteria they have biofilm matrix and it connected with each other. Mm, yeah, here that gram negative bacteria is adjoined to each other cells, and it make capsule coating. Yeah, this is the higher magnifications of gram positive bacteria. This bacteria cell wall and cell membranes clearly indicate that cell walls is spread with external matrix. It enhance biofilm formations. Similarly, that gram negative bacteria, yeah, they have clear cell inner and outer cell membrane with matrix nucleotides. Here, yeah, these all are the biofilm formations and spindle growth. Yeah, according this scenario, we need to develop alternative drug sources, especially antibiotic uses has been restricted or regulated for treating animals. It rise for human consumption with success. Mm, because there are few alternative to antibiotic in case of an extensively resistance or infections. Therefore, it is necessary to develop new therapeutic drugs from source of natural traditional uh, medicinal plants sources. Yeah, here I chose Nilavembu Kudinir Choranam. It have a lot of medicinal property. That Nilavembu Choranam is one such Indian Siddha herbal medicine that has numerous immune and enhancing properties, especially antipyretic, digestive, hepatoprotective, anti-inflammatory, and antiviral, etc. It has been used and traditional remedy for treating various ailment in the Ayurvedic and Siddha system. It also it has used to treatment of different types of intermediate fevers, especially malarial fever, dengue fever, chronic fever, and chikungunya. In recently COVID pandemic, Indian government they prescribed for um, and decoction formulations. Yeah, these are the compositions. These are the compositions of uh, Nilavembu Kudinir Choranam. Uh, Especially this powder present in nine herbal plant ingredient, s- such as Nilavembu, Vetriver, um, Flemichaiver, Mustak, white sandalwood, dry ginger, black pepper, snake guard, uh, parpadagam. These steps are we followed for Nilavembu Kudinir Choranam decoction preparations. Yeah, this is the preliminary studies of bacterial zone of inhibitions in Nilavembu Choranam decoction and standard antibiotics. It mean penicillin and streptomycin solutions. This i- this figure shows an antimicrobial activity of polyherbal decoctions and streptomycin against antibiotic resistance and non-resistant bacterial zone of inhibitions. These white dots circles indicate that inhibition zone of Nilavembu detection and penicillin streptomycin treatment. Compared to antibiotic resistant non-antibiotic resistant antibiotic resistant bacterial strain um, had less zone of inhibition. Mm, uh, but standard antibiotic penicillin streptomycin treatment, the zone inhibitions almost similar with antibiotic resistant and non-resistant bacterial colonies. So we need to develop some biomedical formulations, especially and nanoparticles, mm, because that nanotechnology have attracted and increasing attention in their applications in biomedical engineering, especially in the development of new drug delivery system with help to nano carriers. It transmit drug can reach a specific unhealthy zones. It use prolonged therapeutic efficacy while decreasing undesired side effects. In addition, recent nanotechnological advances such as surface stabilization and responsive functionalization have also significantly improved the targeting capacity and therapeutic efficacy of a nanocarrier assigned drug delivery system. This compared to other nanoparticles, gold nanoparticle have different unique properties. Especially, it is kidney size particle, and it have different shapes also, and it have more surface coating and functionalization groups. So we are used synthesis and characterization of Nilavembu Choornam-gold nanoparticles. Uh, this slide shows synthesis of gold nanoparticles. Yeah, this figure here shows the NC powder, Nilavembu powder extract was boiled and filtered decoctions. That A1 and A2 shows the optimizations of NC extract using different volumes. Here we used 10 microliters as well as 2.5 microliters. That 10 microliter NCT decoction had more turbid and it's not clear visible solutions. Uh, After that, we reduce that volume. Then we use 2.5 ml of Nilavembu particle. It extract it make a clear visible nanoparticles. That B1 and B2 that shows UV visible absorption spectral analysis of and optimized Nilavembu extract decoction as well as synthesized Nilavembu gold nanoparticles. That gold nanoparticle, it have displayed intense peak of at 534 nanometer. It confirming the formations of gold eye transparent solutions. Yeah, this slide shows functional characterizations of NC gold nanoparticles. It used zeta potential and transmit electron microscope analysis to confirm the particle size approximately 56.7 nanometer. It and particle distribution. It's appear spherical shapes particles. Yeah, this slide shows characterizations of NC gold nanoparticles. We used FTIR and XRD analysis confirm the functional group present in the gold nanoparticle. Especially this short peak indicate the different functional group present in the gold nanoparticles. Yeah, according this characterization, gold nanoparticle characterization, we need to move synergetic effect of Nilavembu Choornam-gold nanoparticle and antibiotic-resistant bacterial susceptibility. Yeah, this slide shows antibacterial susceptibility of NC gold nanoparticles. Mm, yeah, figure A and B shows disk diffusion assays at different concentrations of NC-GNP against different bacterial strains. Yeah, here we used four different concentrations of synthesized nanoparticle. Uh, Here, that antibiotic resistant and non-resistant significantly increased zone of inhibition uh, um, in both bacterial colonies. Similarly, we compared with synthesis nanoparticle versus standard antibiotics, penicillin, streptomycin solution. Here, this graph shows uh, synthesis nanoparticle almost more or less similar the zone of inhibitions. Yeah, this slide shows ultrastructure images of resistant antibiotic resistant bacteria strains, gram negative and gram positive bacteria. That gram negative bacteria have rod shape with cytoplasm and plasma membranes. Mm, uh, yeah. Uh, Similarly, that gram positive bacteria they have more dense and biofilm matrix. Yeah, when we treated with NC gold nanoparticle, it may cause cell membrane degeneration as well as chromosomal cytoplasmic um, breakdown. Yeah, similarly, here see these dots all are the gold nanoparticles that gram negative bacteria it directly penetrate into cytosol, but gram positive bacteria it bound around the mm, cell walls. It means that gold nanoparticle have negative charges. That negative charges interact with gram positive bacteria. Similarly, that an this image shows ultrastructure images of non-resistant bacteria saying gram negative and gram positive bacteria. Here also that NCGNP treated bacterium uh, had breakdown and degenerations of cell wall and cytocells. Here, this gram positive bacteria it's mainly adjuvant surface of the cell walls. Yeah, this image high magnification images of antibiotic resistant gram negative and gram positive bacteria. Here it's clearly indicate that gram negative bacteria have outer and inner cell membrane and nuclear content. This gram positive bacteria have thick cell wall and cell membrane with nuclear content. When treated with gold nanopart NCGNP, that gram negative bacteria. It penetrate inside the nucleus, and it make vacuum, cytosol vacuums. And gram positive bacteria, it adjuvant surface of the cell walls, and it make uh, degenerations. Yeah, according this overall scenario, this overall summary represent the polyherbal plant extract of Nilavembu choranum was successfully used to synthesize gold nanoparticles through a plant extract mediated green synthesis method. This Nilavembu choranum decoction was found to contain various phytoconstituents that were responsible for the formations and stability of gold nanoparticles. Mm, Further, UV visible spectroscopy, FTIR, TEM, DLS, and the XRD results reveal the size, morphology, crystal structure, and stability of gold nanoparticles. When compared to Nilavembu extract, the synthesized NC gold nanoparticles had effective antibacterial effect on both antibiotic resistant gram positive and gram negative bacteria. Although the present scenario significantly increased increased therapeutic efficacy on antibiotic resistant as well as antibiotic non-resistant bacterial infections. So the possible concerns: gold nanoparticle is safer. Also, Nilavembu Choranum extract formulated gold nanoparticle was synthesized in an e- um, eco-friendly and cost-effective manner. It biologically inert due to less particle size. It won't interact with other toxic chemicals and adverse effect. Mm, eh, this study results indicate that the NCGNP treatment inhibit microbial adaptation and biofilm formation. In future, this therapeutic efficacy result beneficial to sterilization and reduce co-infection mortality. Yeah. Uh, uh, this present study recently we published in February, end of February 2024. It is in online. Thank you so much for your kind attention. If you have any question, please discuss, convey me. Thank you so much.
Thank you, sir.
Yeah.
That was a highly informative session. As per your
Yeah.
research, it clearly shows that we can use gold nanoparticle synthesis to nilavimbuchuranum as antimicrobial agent.
Yeah.
And there is one question in this.
Yes.
When you're synthesizing gold nanoparticle, being a metal, how it will get excrete from our system? Will it damage the system? Will it
Because
damage
compared,
kidney or anything?
yeah, yeah, compared to other nanoparticles, that gold nanoparticle have basically have more biological safer properties. That's why even when we use sweets and other uh, food, those food we always mixed with gold films, because it is safer and it's make some protective properties. It won't deposit inside the organs. It excrete very fast, simultaneously.
Easily, okay. As you
Yes,
said, it
yes.
excreted easily from our system. As per your result, it is clearly shows that we can use gold nanoparticle synthesized Nilavembu as an antimicrobial agent in future. Thank you, sir.
So
That
not
was an
only,
excellent presentation.
yeah, yeah,
Ah,
not only antibiotic agent, it is concern sterilization and recovering the post-COVID morbidity and secondary infections.
Oh, okay, okay. Okay, sir, that was nice work presented by you. Thank you, sir. And
Yes.
is there any other question from the audience side? Okay, sir. Thank you, sir. Actually, the session was so clear. Thank you, sir. On
Okay.
behalf of
Thank
the
you so
organizing
much for.
committee, uh,
Yeah.
on
Okay.
behalf of
Thank
the
you so much
organizing
for.
committee, sir, shall I
Yes. Yes.
complete?
Yes. Yeah. Yeah. You complete.
On behalf of the organizing committee and all attendees of the session, I want to extend our sincerest gratitude for your captivating and enlightening presentation at our international conference. Your expertise and insights truly elevated the session and left a profound impact on everyone present. Your ability to engage the audience and deliver such valuable content in a testament to your remarkable skill and knowledge in clinical science and nanotechnology. We are deeply grateful for your contribution to make our conference a resounding success. Your willingness to share your expertise with us is greatly appreciated and has undoubtedly enriched the experience of all attendees. Thank you once again for your invaluable contribution. We look forward the possibility of collaborating with you in future and to continuing the exchange of ideas and knowledge. Thank you for joining us today. Thank you, sir.
Okay. Thank you so much for giving this opportunity. On and all, I thank all the organized team and chairpersons. Thank you so much.
Thank you, sir.
Bye
Thank
bye.
you so much. Bye bye, sir.